Hello and welcome to this video where we will expand on the use and purpose of immortalized cells. Having heard the name, you might think of something like a cell that cannot die, but that is far from the mark. Immortalized cells are generally a sample of cancer cells that proliferate without any pause or control. A kind of immortality, but more in line with Dorian Gray than Jack Harkness. As a tool for research, immortalized cells are a massive boon. These are cells with a relatively consistent cell type that, once the genome is modified, can be cultured to produce a large, viable, and stable sample. By contrast, trying this with a cell line that lacks the attributes of an immortal line would lead to random, spontaneous mutations and unreliable experimental results. Compared to your primary cells, this allows for replicability between studies and research institutes. The downside to it is that immortalized cells come from a pathological state. You cannot extrapolate their findings effectively or reliably to a non-disease state in all cases. Conversely, your primary cells, which are generally of a non-pathogenic state, can be neither replicated or cultured continuously, nor can they be as easily compared between studies due to the relative incidence of mutation within them. Not all immortalized cells are cancer cells, but it is a prevailing source. Others are derived from stem cells or custom created immortalized cell lines. Immortalized cell lines have undergone similar mutations, allowing a cell type which would normally not be able to divide to be proliferated in vitro. This means they can continuously be cultured with a consistent genome between generations and for an extended period of time. Primary cells can only be passaged or cultured so many times before they begin to mutate and deviate too far from the original stock to be considered useful. Immortalized cell lines are used as a very simple model for complex biological systems and disease states. Because of their nature and that they are, by their terminology, a pathological sample in many cases, they are used primarily for biochemistry and genomics. You can get a lot of useful information by looking at the chemistry within the cell and the genetics underlying the processes that occur within them by using this particular cell type. They're not just a research tool. Immortalized cells find an ever-increasing role in biotechnology. Think of it this way: they are able to be genetically modified to create a pool of common cells that all have the same background and function. You can continuously culture them to create more. They won't change by much, and they will therefore continue to function the way you want them to. You can take these cells and modify them to create a variety of products. This could be something like platelets, new erythrocytes or red blood cells, and more. You not only need to consider their role in production, but also in translating biotechnology from the drawing board to reality. Animal models are frequently used, but they also fail to communicate failures in a human in an animal model. There have been many examples of where mouse, rat, or ape works, but once you put a drug into a human, there are unanticipated and very undesirable side effects. Using these cells, which could be from a human and immortalized, allows for a cheap and effective way of testing the safety for specific cell types, and by extension, the safety of certain tissues within the human body. Getting your immortalized cells is interesting, as there are a number of ways to get there. One of the methods is simply isolating them from a naturally occurring source. This is nearly always cancer. If not cancer, then certainly a tumor. Both of these are defined by their characteristic division and replication without stopping. Then you have examples of this, particularly the HeLa cell line, which came from cervical cancer. After this, you have those cells that were made to mutate, whether that's through a virus or a chemical. Finally, you have those that have been artificially selected for and or modified. These cover the key ways of getting your immortalized cells, and knowing what has been said so far, you are probably thinking that this is all well and good, but you could learn as much from Wikipedia. Naturally, this is not the sum of what the video is about. What we wish to add onto this background is how and why we can genetically modify these cells for good and interesting reasons. The simplest way to create a new cell line is to modify one that already exists with close to or related problems or objectives. If you're looking at immortalizing your cells, you could look at taking a primary cell line and then changing it. The easiest way to do this is to look at what cells you are already looking at, have access to, or might be able to modify that are optimized for the growth and duplicational culturing of viruses. This will maximize the recombinant protein production, a significant feature for viruses, as they must be able to replicate at a significant rate to then survive and find a new host once their current host is destroyed. The creation of a completely new cell line is difficult. It requires the use of cutting-edge technology, raw materials, time, expertise, all the things that cost a researcher essential resources they need for their research. Finding a way to accelerate this is key to any successful development of a cell line. That's why we've been looking at viral infections. Viral infections are especially useful as it will turn the cell into a tumor. Bear in mind that not all cells exposed to a virus become a tumor, but neither are they unharmed by the process. There are specific oncogenes that have a number of layers of security on them that are designed to prevent the cell from continuing to replicate continuously. But the viruses have, in evolutionary terms, adapted to this and found ways around those. One of these is a gene called human telomerase reverse transcriptase, or HTERT. This can be used instead of a viral gene. Modifying it or altering it can produce cells that behave like a primary cell line, but will propagate continuously like an immortalized cell line. This gives you the best of both worlds. Better yet, the method is well enough established that there are commercial kits available where you can order it in, match it to your cell line, and with the appropriate knowledge base, create immortalized cells in a primary cell format. This avoids many of the issues around sourcing and using immortalized cells. An example of this involved a research team looking at bats and coronaviruses. The research team was able to source 3 different cell lines from 3 different bat species from which they could culture novel coronaviruses. Remembering, viruses sometimes need specialized cell lines, and without them, they simply will not grow. Another method available and has been used effectively involves chemically altering the cell. One of these used dimethyl sulfoxide to change the regulation of CMYC, an oncogene. Changing this allowed for differentiation of erythroid cells from stem cells and progenitors that otherwise were not going to begin differentiating. This allowed them to look at bone marrow stromal cells and create a vast array of hematopoietic cells. These were then used for immunological studies. It included dendritic cells, mast cells, and natural killer cells. It could then be expanded and was expanded to include vascular smooth muscle cells, blood vascular and lymphatic endothelial cells, tendon cells, Sertoli and Leydig cells, and blood placental barrier cells. So far, we have touched on 3 methods: finding a immortalized cell line by luck, playing with viruses and or the genetic code that they are affecting, and interfering with the cell's genetics using chemicals. These various methods have allowed the development of novel strains of animals from them. One of these is known as the immortal mouse. It has cells that can continually produce immortal cells from a wide variety of tissues. Having done that and found an animal model that could survive to some extent, they are able to then create other models of disease, and this could be important for investigation of cancers. Cancer is not the only way and place in which this can and should be used. Immortalized cells begin to differentiate and continue to differentiate. This means you can look at a cell that has multiple generations or processes involved at which it will differentiate into different kinds of cells, such as those in the hematopoietic tree, and understand what signals are needed at what stages to produce specific final products. This knowledge has given rise to something called the Reneuron in 1997. It was a project with the objective of developing a novel human cell line for transplantation. Being a Reneuron, they were looking to transplant neurons. The idea was to get neurons that they could transplant into patients to act as brain cells and reverse damage to brain tissue caused by stroke. That would be a huge achievement if it can get there, but the problem has always been the cell growth and the screening needed to ensure they're selecting for the appropriate cells to go in there. Then we also have to consider the ethics. The possible use of stem cell therapies for conditions such as stroke, where few to no other treatments exist, is fraught with problems relating to whether or not patients should be given something experimental like this, what the benefit for them is, and whether or not the risks outweigh that benefit. We can then look at more fundamental production requirements. There are 2 parts to this. One is an example in which they have used immortalized cell lines derived from primary chicken embryonic fibroblasts. These act as a substrate for developing and growing viruses. Propagation of the virus is useful and important if you're looking to create something like an influenza vaccine. The specific substrate chosen is a powerful tool for using as it is because it does not require the mass collection and utilization of whole eggs, which has some considerably greater ethical concerns behind it, but it also allows for a more efficient and safer production. To a certain extent, you can take this idea and expand it to look at the meatless meat burger. Myocytes are a kind of cell. Specifically, they are the cell in muscle that allows it to contract. They are also the primary thing we eat when we are thinking of meat. If you were to try and propagate the muscle cells as they are, you would find the same problem that has been found with all primary cells. They cannot be reliably propagated without eventually mutating or failing. In order to make them work properly, you would have to immortalize them, and that would allow, to some extent, for the production of meat without needing to actually make meat in the terms of an animal. Taking the concepts we've mentioned so far, the next obvious development should be obvious. The production of blood to meet the increasing worldwide demand for a safe, reliable supply. Red blood cells, and specifically the type O negative, are generally in short supply across the world. There are a few people with that blood type compared to the others, but they are considered the universal donor. If you could look at that hematopoietic tree, find the right way and the right cells to eventually get to a type O negative red blood cell, you could drastically bolster the supply with a stable, safe source of red blood cells. There's also possible options to extend this to cord blood. However, we're not yet at that point. What we are at is the production of at least progenitor hematopoietic stem cells, which could be given to otherwise healthy adults in need of blood. They would then differentiate efficiently, effectively, and reliably. These could then be filtered if need be, or they can be given to the patient once they've differentiated into the next stage, where they should enter to some extent of a normal cycle. Even if we weren't looking to use this for acute care of trauma patients, there is possible application for those with a chronic disease, such as sickle cell anemia. Finally, there is the production of skin or skin equivalents using immortalized cell lines. Human keratinocytes could be taken, and they have been in the past, to create immortal cell lines and used to generate a sort of human skin that could be put over burn victims' damaged tissue. This would then be an acceptable graft that could at least somewhat mitigate the risk associated with skin grafts. These are the possible beneficial applications. We will later look at the negative applications of immortal cells and give a greater focus to the problems and deficits of immortalized cell lines. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.